Okay, it's recording now.
Yeah, we went there and it felt like
Whenever you're ready.
but we, it's been one of the nice things there, like going on bike rides. There's all these sort of country restaurants that feel stuff. It's not as nice to do.
I'll stay for a few minutes and make sure.
Okay.
it's not allowed to talk.
The weather was good.
I don't break anything.
Good outside.
Where in Spain?
Malaga,
Hello, everyone.
our good friends.
Thank you very much for being here. Today, we have Dr. Ksenia Krasileva to share her recent research. Dr. Krasileva is an assistant professor of genomics, computational biology, and plant immunity at UC Berkeley. She did her PhD with Dr. Brian was at UC Berkeley, who is one of the fathers of the molecular plant pathology. She did her postdoc with Dr. George Dubsworski at UC Davis, who is a wheat breeder. Then Dr. Glasgow built her own lab at the Earnham Institute and the Sainsbury Laboratory, Norwich, UK, where I met her in person for the first time. So excited. In 2018, she moved back to UC Berkeley. Dr. Krasileva's group is part of the Department of Plant and Microbial Biology, the Innovative Genomics Institute, and the Center for Computational Biology at UC Berkeley. Her group uses the latest technologies such as sequencing, synthetic biology, genome editing, and computational modeling to study genome evolution, plant innate immune system, and pathogen virulence variation. So impressive. They use several host pathogen systems for study, including wheat strip rust pathogen and the rice blast pathogen. So everyone, please put your hands together for Dr. Krasileva. Thank you. Thank you.
All right, thank you, Wei, for inviting me here and a very nice introduction. Now I have to live up to the energy of the room and present the research. I'm going to talk today about 3 different stories that probably represent the breadth of the work that we do in the lab. If you have questions about anything else, please reach out by me by email. If you have questions about these stories, also always feel free to reach out by email. Before I begin, I do need to tell you that it's actually a very difficult time for me right now because I'm a Russian American, and I want to make a stance for no war in Ukraine, alongside with very, very few and very, very brave people who are making that stance right now inside Russia, including like my hometown, St. Petersburg. So if you all have any Russian or Ukrainian friends or colleagues, do reach out to them with words of support right now. We need it. Thank you. Then I want to tell you a little bit, I want to put an acknowledgement slide and tell you about the absolutely fantastic team that is doing the research that I'm, traveling and presenting and have a privilege of advising. And here's the group in 2022 in the Silomar on our lab retreat. And the work that I'm going to tell you today is going to be has been done by a very talented graduate student, Kun Yang Seong, who brought a completely fresh new perspective to how we do comparative genomics to the lab using computational structural biology. Also, I'll tell you the work done by Erin Baggs, who had been a first PhD student in my lab and now is staying for a short postdoc to finish up the absolutely magnificent research program that she started. Our long-term collaborator is Daniel Prigozhin, who is a structural biologist at LBNL and also an immunologist by training. And a couple of former people, a postdoc Irina Tamborsky, who contributed to some of the engineering work that I'll describe, and an undergraduate, Mark Tiesma, who contributed, who was mentored by Aaron and is now actually applying to grad schools. So I have a couple of undergrads applying to grad schools. If you have particular programs across campus that, let me know. I'm trying to advise them on where to go and which PIs to work with. So, so they did the work. We are part of the Center for Computational Biology. Our department is plant and microbial biology, and also part of the Innovative Genomics Institute that's mostly focused on genome editing. What we provide is basically more of a computational perspective of what are the targets they are working on developing the tech and doing the editing. The research projects in general that we work on, they span 3 different areas. Most of it is built on our understanding of natural diversity, and that's the bread and butter for our projects. We're also starting to leverage induced diversity and synthetic biology. And for those of you who are grad students and postdocs, this was not accessible at the time when I was a PhD at that rate that we have now. This is completely revolutionary now. You're not bound what you can do. You're only bound by your imagination, what you can synthesize. But gene synthesis is cheap enough that you can do anything now. And we take our inspirations from understanding the natural diversity. And the main paradigm that drives, I would say, all research topics in the lab is how can organisms, and especially plants, that only have innate immune systems, so germline encoded sets of immune receptors, compete and evolutionary be able to recognize diversity of pathogens, rapidly evolving pathogens that have, the same diversity of disease-causing molecules that pathogens that impact us with adaptive immune system, right? So how is it possible? And to summarize quickly in 2 slides as a background, 30 years research on the molecular plant pathology topic that you just need like a little bit to understand what we do. It comes down to that fact that many different pathogens, when they infect their host, they infect a plant cell, no matter what type of pathogen it is, what kingdom they come from, they inject their own effector molecules, proteins, inside the plant cell. They secrete them outside of the plant cell. And the role of these effectors is to establish disease, is to suppress immune system, acquire nutrients. And those effectors is what actually reveals to the plant immune system the presence of the pathogens. And plants can really differentiate that. And we talk about the sets of extracellular immune receptors that you can recognize pathogen-derived molecules. And my lab specializes in the sets of immune receptors that are inside the cell. And they can recognize this pathogen effector molecules, either directly binding them, right? Or monitoring this host proteins targeted by the effectors. We've also described a phenomenon where the homologue of these targeted proteins can be integrated in and become part of the immune receptor, making it a single module system that with a bait for the pathogen. And there are those immune receptors that don't serve the sensory function, but their signal transduction function, they're amplifying activation of the immune system. All right. With that in mind, in order for us to understand how can plants efficiently utilize these immune receptors to recognize the diversity of pathogen molecules, we also want to understand, how diverse those pathogen molecules really are, right? And what we know is there isn't any rule of what a pathogen needs to become a successful pathogen. it can be a virus and only utilize very few genes to successfully infect the host. Or if it's your fungal or mycetic pathogen, you can literally have hundreds and hundreds and then thousands secreted proteins that are presented to the host. So in our mind, we always thought, well, how do we begin to make sense of it? And especially, how do we begin to make sense of pathogens that can encode hundreds and hundreds of effector proteins that are very commonly unrelated in their primary sequence. So they're hard to understand are there any similarities across pathogens or even within pathogens in this effectors. And my first message from the talk that I think nothing in evolution makes sense except in the light of structural biology. So what we decided to do, and by we, in one way, I mean, it's a graduate in Kunyang who came to me and said, Ksenia, we've been studying these effector proteins and solving structures of it for years. And I can say, I solved one structure, one effector together with another graduate student. So it's a 2 students, 6-year PhDs, one structure, right, for one pathogen. So the process of experimentally solving the structure has been so slow. However, the patterns emerged. We were able to observe that sequence unrelated effectors across some of the pathogens, they share the same holes. And the student who came to me, Kunyang, he said, well, Ksenia, you know what? And he came to me in 2019, I think. He said, you know what? There has been a revolution in how we can predict structures. So let's do a computational approach, computational structural genomics approach, to take all the secreted proteins, predict their structures, and then see who's similar, what are these commonalities. And the revolution that he talked about was relatively unrecognized, except for the field of structural biology and computational biology in 2018, but it gained much more attention, of course, in the recent years. And it was availability of new algorithms such as TI Rosetta, developed by David Baker's lab, who's been working in this field for many, like 20 years, and AlphaFold 2, developed by DeepMind, now owned by Google, that is able to take a primary sequence, and take a coevolutionary information about the sequence from all of the databases, and take databases of known protein structures, and get training in the algorithm good enough that it can then take a new primary sequence and predict how it folds. Essentially transfers this conformational landscape and find the lowest energy state. And this is a super hard problem computationally to solve. So the trick was to train it based on this coevolutionary data that residues are co-evolving, they're more likely to touch each other in 3D space, basically. But you don't need that information to predict the structure now. You can have a primary sequence, no homologs. There is still algorithms that are trained enough to do this, relative different success rates. So, and at the time, again, we were starting to do that like 2019. That seemed like magic, right? Really? Is that possible? How good is it, right? So what the first thing that Kunyang did, he did a test. And he did a test that was a standard in that computational structural biology community. He took effectors because we wanted to know how good these programs were on the effectors. There was a special, we think we would like them, we study them. And he took effectors across fungal pathogens for which experimental structures have been solved. And those included several that had solved structures that were not part of the databases used to train algorithms. That's important. And he ran the prediction software, and then he could compare the model that the software predicts to the actual true, to the experimental answer. And this is what the actual precision score is. where one, those models completely overlaps, the amino acid side chains are in the right places. 0.5, the fold is correct, right? The overall fold is correct. Amino acid side chains might be going in different directions. Below 0.5, well, it gets wrong, right? The fold is wrong. And what you see is basically there is a substantial number of effector proteins for which we could get the correct fold. Now, what was more important than that is the correlation between actual precision and estimated precision. Estimated precision is produced by the prediction software. It's their estimate how they think their model is good, how good their model is, without knowing the true answer, right? Because for most of the effectors, most of the structures, there wouldn't be a true answer. So we have to rely on some sort of metric to say, well, is that a good model or not, right? To do some sort of any meaningful analysis. And what you can see is that we reproduced what was shown for this programs for benchmark sets of proteins, that estimated precision has a linear correlation with the actual precision. So we could trust estimated precision scores well enough to exclude poor models and then only take forward our analysis on models that we think would be relatively good enough. Okay. So now the question became, can we use computational structural genomics on a genome-wide scale to identify sequence unrelated, structurally similar, we call them sus, effectors. And so Kunyang installed Rosetta first. AlphaFold 2 was not available at that time yet. And he took the first fungal pathogen that was ranked as the top one out of top 10 fungal pathogens of plants. This is the top one, top threat to our pathogens by the community, by the molecular plant took the review, took the top one. He took 1,000 secreted proteins, predicted secreted proteins from that pathogen called Manopor. He ran structure prediction on our cluster. He installed all the software, ran it, and he was able to see that. And then he wrote algorithms to cluster the structures, compare the structures and cluster them, that he could indeed identify sequence unrelated. So on sequence level, there would be 6 different clusters, effectors that have the same fold. So the approach worked, and out of 1,000 secreted proteins, we actually got about 150 structural groups in this regard, and about 75% of these 1,000 proteins had models with estimated precision above 0.5. So not full success, right? Not every structure can be predicted, but it was good enough to continue. What he then did next, he said, well, let's now move on to comparative computational structural genomics. And at that point, he switched to AlphaFold 2, again, installed it on the cluster, and ran about 26 different fungi, secreted proteins from 26 different fungi that included both pathogens and their close non-pathogenic relatives, as well as some yeast and oomycete that's unrelated to fungi. To answer 2 main questions that were really interesting to us. Are pathogens going to share structures of effectors, right? Is there a particular fold that makes a pathogen, a plant pathogen? Sets of folds. Are they going to also share folds non-pathogens? Can we start to evolutionary untangle where the pathogen virulence factors, these effectors come from, originate? And by sequence similarity, we cannot, actually, a lot of times. So the results of it was that indeed, if we took all of these structures, and that's about 22,000 predicted structures across all of these phytopathogens and their relatives, we can see that First, we saw slightly variable success rates, again, in how good the programs were able to predict the secretomes. I'm happy to talk about it separately. But we saw that many folds were shared across pathogens and non-pathogens, and also many folds were highly expanded in pathogens. But what I'm going to show you next is that there isn't one rule how a pathogen can become a pathogen. Different fungal pathogens have slightly different sets of highly expanded folds. So what does this data actually look like? Well, we organize it we represent the tables in plots like this, where you have your fungal species on the top. This is structure clusters, and they're ranked here in size. So the largest group of proteins includes proteins shared across many different fungal species. It's actually a yeast killer toxin-like fold. And so these sets of proteins that what I'm showing you, the non-pathogens have these folds too. They do include representatives of known effectors, so known virulence factors. What these told us that most likely then a lot of effectors, they're not generated de novo from the genomes, right? They are actually derived from secreted proteins in non-pathogenic species. So what happens to them? What we were able to tell, and here each dot is a sequence similar, is a cluster of sequences based on sequence similarity, and the links between these dots is a structural similarity. So there's a particular cluster that's shared across pathogens and non-pathogens, and it has links to a cluster that's highly expanded in a particular pathogen. So what we think is happening, there is a secreted protein with probably ancestral function that is undergoing very rapid expansion and sequence divergence and becomes co-opted for the pathogenic functions. Theory is quite simple. We never had evidence before for that is actually what's happening. Now, other groups of proteins, and so if you follow that trajectory, you see that these links become weaker and weaker, right? Even in the structural space. So what we think happens next is this similarity at some point even become lost. So we have classes and groups of structurally related proteins again that are now becoming pathogen specific. And so different pathogens have expanded sets of particular folds. Blumeria graminis, and that's a really nice example. It has about 600 secreted or 800 secreted proteins. About 400 of these have RNase-like fold, same fold. And so on, and different pathogens utilize that too. So it becomes we're now starting to answer that question. There isn't one way how a non-pathogenic fungus can become a pathogenic fungus. There are different evolutionary, you can use basically different folds, but then achieve the same goal of targeting plant proteins to get nutrients, suppress immune system, cause disease. Quite interesting, right? So the actual ancestral function of that secreted protein actually becomes irrelevant. A lot of time we see loss of even catalytic sites. And this RNase-like effectors, which are known effectors, known virulence factors, the catalytic sites for RNase activity is gone. It's the surface of the protein, their ability to bind and modify plant protein that starts to matter. In one of the most interesting examples, and from Ustulagomatis, what we saw is that when we took the structurally similar clusters, they actually form an array in the genome. So previously it was known that Ustulago has a couple of known effectors that, including this here at TN2, and their neighboring genes that actually don't look much like this known effectors. If you look at these now in the structure space, it's an array of structurally similar proteins. So again, supporting that, in this case, there's probably local duplication making this array. And there's such a rapid divergence that on this primary sequence level, we basically stop seeing sequence similarity. On a structure level, we still see this belong together. Okay, I have a couple more stories. If I don't get through all three, I'll just tell you two stories, okay, just to leave room for questions. But to summarize this, what we think is happening, how a pathogen is derived from a non-pathogen, there is probably an ancestral secreted protein and its friends in the sequence space that then undergoing very rapid divergence and duplications. Some of them might be lost by drift and loss of function if, for example, they're being recognized by a plant. And then there is a functional, and we have evidence for transcriptional diversification among them to make it a more successful pathogen, to change host specificity, et cetera, et cetera. All right. I'm going to move on. How you usually do it? Do you take questions after, like, there's a first story questions, or do you want it questions at the end?
However you like.
You what?
However you like. Usually we do it at the end.
Usually you do it at the end. Are there any burning questions right now at this point? No. Yeah, Haley.
Very exciting story. I just wondering, later on the structure was confirmed by the AlphaFold 2.
This is now AlphaFold 2, yeah.
So these two algorithms give you very similar structure
Yes.
prediction.
Yes.
Overlap
yeah.
very well.
AlphaFold 2 gives you, and we did direct comparison on Magna Portha. It basically, they don't differ which structures they can predict correctly on the fold level. AlphaFold 2 gives you a slightly more precise placement of side chains, amino acid side chains. For our analysis, it doesn't actually matter because we are operating on the fold level. But mostly they do, yeah. What I think what AlphaFold DeepMind people did, they took David Baker's work and they improved it using their algorithm because they have just a better algorithm. But it's built on like 20 years of work, no public.
Great. Did you find any effector proteins from fungi that have structural similarity to RXLR1? You listed it here, but I don't know.
No, our XLRs are all MySit specific. So this is an example of, this is the pathogen becomes a pathogen specific. Yep. Good questions. Okay. Now ready for the next one. So kind of the next paradigm, okay, we can start to understand the space, evolutionary space of the disease-causing molecules. Now let's look at the plant side. And what happens in plants is in wild populations, you have a lot of diversity in immune receptors on a population level, even within single species. What we done through breeding and planting monocultures, we undermine the fundamental principle of a functional immune system, any type of functional immune system. We got rid of that diversity. We're basically planting plants with same sets, exactly the same set of immune receptors. So if a pathogen overcomes it, it can wipe out the whole population. And so we started to understand that, well, what are the different ways we can look at it? And Aaron had a very fantastic idea. Said, well, let's look at the natural diversity in copy number variation of these immune receptors. And she found several, looking at biodiversity and copy number variation of immune receptors across all sequence plants that she could do at the time. She found several clades, including duckweed, that naturally lost most of these NLR immune receptors, right? I just told you this is our gold mine in the field. This is the disease-resistant genes. Yet there's an invasive species clonally propagating that doing just fine without them. So how? That became Aaron's PhD topic. And to give you a perspective, a wheat variety can have up to 3,000 immune receptors. If all of them are the same, right, and you plant a monoculture, they don't help. If they are overcome by the pathogen, if there isn't one of them that's being able to recognize the pathogen, you have disease. So duckweed told us a couple of things, and what here I'm showing you is a simplified now version of the comparative genomic analysis that Aaron did, and she compared plants from monocots and dicots to several duckweed lineages, and duckweed is a monocot, so it's more closely related to rice or wheat than to Rhodopsis or tomato. And she observed reduction in these immune receptors, both the extracellular and intracellular one in duckweeds. She observed complete loss of signaling pathways that these receptors use. So that's gone. And then she observed an expansion of antimicrobial proteins, specifically of this MIAMP type. So Rhodopsis has 0 of these, tomato has 0 of these, rice has 0 of these, duckweed have 50. So one of our hypotheses is that it's the duckweeds lost NLRs, lost that signaling. We don't know why, whether it was detrimental to have it, but they compensated it by expansion of another protein family that can do the job of keeping microbes at bay. What Erin did then, she did something very brave. She went to the lab, she got duckweed. She first ordered it on Amazon and said, hey, Ksenia, I really want to grow duckweed in the lab. So I just ordered it on Amazon. I want to put some pathogens on top. So I'm like, well, Erin, fine, but let's get like a lab strain of duckweed. So I emailed around, and close by at J-Bay, Jenny Mortimer had duckweed. And I was like, okay, I brought duckweed, and this is what duckweed looks like. You can grow it on plates. And then Erin started basically vacuum infiltrating or flooding duckweed with various bacterial pathogens from our crop species, Pseudomonas and Anthomonas. And quickly enough, just says maybe duckweed figured out something, that it can resist all the pathogens. But actually, this generalist pathogens can infect duckweed, cause disease and infection. But what we got now, we have a novel pathosystem. So a duckweed Pseudomonas pathosystem didn't exist before. Now we have it. And so Erin started to characterize it. So what is actually happening during this interaction? One of the things, we don't have fully the genetics of duckweed. There are labs working on it. We wish we did. But what we do have, we have the genetics of the pathogens. And we have very much a lot of information of Pseudomonas being used on the Rhodopsis for years. And so very quickly, what Erin did, she was able to use a type 3 mutant of Pseudomonas, and that's a secretion system mutant that would deliver effectors inside the plant cell. And that mutant line could not cause the disease. So we now know that the disease of duckweed by Pseudomonas is caused by effectors. What Erin also did, she did an RNA-seq analysis. And the most upregulated genes that she found was actually those antimicrobial peptide proteins. So together, we think that these antimicrobial proteins may play a role, at least they're triggered by infection. They're not, of course, sufficient, right, to completely stop all the pathogens. What we're doing right now, Erin picked and synthesized about 15 of these proteins, and we're throwing various fungi and bacteria on them to see if they actually have antimicrobial activities. The big picture takeaway is we went from our looking under street lamp, right, to look at canonical immune pathways, surveyed the biodiversity, and now the biodiversity in duckweed told us that plants can do things differently. And I think, well, one way is we think is this antimicrobial proteins. We'll see if it holds. So far, it all points to it. But then the other bit that Erin did is like, well, but in the wild, in the pond, duckweeds are surrounded by Pseudomonas. Pseudomonas is ubiquitous. It's in rainwater. It's everywhere. Yet, are they sick, right? They're not. They're invasive. So what is going on? And the next thing she did, she basically went to UC Berkeley Botanical Gardens. So the next message I'm getting from the students is like, okay, we have all the year, we're gonna go to UC Berkeley Botanical Gardens. They have ponds with duckweed. I'm like, oh, maybe we need a permit. Let me email the director of UC Berkeley Botanical Gardens. I'm emailing the director. We got the permit. They're like, yeah, fine, come sample. Put us in the acknowledgements, please. And so Aaron and a rotation student at the time, Grace Stark, they got the gear, they went, they sampled 3 ponds at UC Berkeley Botanical Gardens that had duckweed in it. They tried to find disease on duckweed, sick duckweed, couldn't find any. All the duckweeds were super healthy. And they took the water from it. They passed the water through the filters with the idea of, well, let's see what microbes are actually there in the pond and which microbes are in the water versus which ones are on the duckweed or inside the duckweed. Thinking of more, maybe it's, their microbiome aspect to it. Now, in one control that so here's comes me as an advisor, besides writing and getting permits and lab strains, I asked them to test if the water that they filtered had any effect on the disease symptoms they would observe for the lab duckweed. Maybe it's a pH, right? Maybe it's, there's something in the water. They did the assay and they used initially 0.2 micron filters. That's what we usually use in the lab. They're the cheapest. And they actually saw that lab duckweed incubated with 0.2 micron filtered water from the pond got the protective effect against the pathogen. There was something in the water, there was something in the filter actually. But then it turned out, and this was again like Aaron searching the literature, if they boiled the water, the effect went away. If they passed that water through 0.1 micron filter, that would pass all the viruses through, but not the tiny bacteria, including some of actually Pseudomonas, the effect went away. So very lucky, we fractionated the water and we had the fraction that can give us idea that there is a protective effect from the water in the natural environment. And it's, it's basically smaller than 0.2 micron, larger than 0.1 micron. So Aaron plated the 0.2 filtrate. There were bacteria in it. She then went and she isolated single strains, and she did co-inoculation assays where she challenged the single strain bacteria isolated from 0.2 micron filtrate from UC Berkeley Botanical Gardens, together with either Bacteria alone, our bed bug, right, the pathogen, on a sterile laboratory strain of duckweed. And what you can see is that some of these isolates actually have different effect to duckweed, some of them may be beneficial, but there were 2 that gave protective effect. So she recapitulated that actually there is something in the water and it is a microbe, and she sequenced those genomes, and both of them turned out to be other Pseudomonas. One of them is a new species. It currently just has a taxon ID. Aaron thought about naming it Pseudomonas achlones after the land of which Botanical Garden stands, but the paperwork required to submit a new species name was insane. So we kind of gave up on it. And there's more data that we have currently, or the kind of the data that we have points out that there's actually direct interaction between these 2 Pseudomonas, and the environmental strain can directly suppress the growth of the Pseudomonas syringae in vitro when they're co-cultured together in the test tube. Whether it's a contact dependent or independent phenomenon, we don't know. But our model is that duckweed antimicrobial peptides probably do play a role, at least for some pathogens, but it's up for testing. But definitely there are friends, and now we've confirmed that the friend actually colonizes the duckweed in the wild, but can go through stomata and establish itself inside duckweed, and it's able to help duckweed to fight the foe. If you to preempt the questions, we tried to save Arabidopsis using the friend. Doesn't work. Arabidopsis doesn't allow the growth and colonization of that bug. So even on like this EDS1 or pathway mutants. All right. How are we doing it with time? We're doing relatively well, right? Any burning questions about duckweed? And I'll leave time. Yeah,
So you showed that there's this expansion of antimicrobial.
Yeah.
And the closest relative that you surveyed was rice. I was wondering if you looked closer.
yeah, we did. It was a reviewer comment in final published version, and actually it's taro is the closest relative, and it does have them, but still not as many as duckweed.
It does taro maintain the MLR.
It's in the table in the paper. It does maintain the NLRs and EDS in the pathway, but to what level I don't remember from the top of my head. We only have again N of one genome of taro. So, and there is nothing in between really. So in terms of biodiversity, I think we can do much more refined sampling as well going forward. All right. Last but not least, something that I did my PhD on, I want to tell the story of actual how NLRs can be helpful to the plants that have them. And I just told you that a functional immune system that relies on NLR diversity in plants depends on the diversity generated in these receptors on the population level. And up until we moved into the pan-genome stage of sequencing, we couldn't really understand this diversity of these immune receptors on the within the species because they have high count of repeats and they highly repetitive. They form this arrays as well of genes. So none of the Illumina short-read sequencing could actually give us a good enough information about these genes. But in 2018-19, and the first pan-NLRome, long-read pan-NLRome of Arabidopsis came out from Detlef Feigel's lab along with others. And then soon after, there were long-read pan-genomes available for several crop species. And so what we said, we developed a workflow to analyze point mutation and natural diversity of these immune receptors by first phylogenetically grouping them. So it's a phylogeny, here's a phylogeny of NLRs from 50 Arabidopsis ecotypes. This is 10,000 genes in this tree. Splitting this phylogeny into the evolutionary related orthogroups or clades, realigning them full length. So we know full length alleles for each gene, for each ortholog from each of these accessions, and doing sequence diversity analysis. This is not a PopGen metric. This is more of a metric used for antibody diversity analysis or also for metagenomics. We're basically walking across the alignment and looking at how many amino acids are present in this particular column, right? And that's the measure of the diversity where, so one is you have only two amino acid sites present, variants present in the column. Above one, it's what's called multi-allelic sites. More than two variants are present in this population. When we combine this data with, again, structural models, we observed that these highly variable residues clustered together on the protein surfaces in the regions of the proteins that are ligand binding. And we postulated that basically what we're observing here is highly variable ligand binding sites, generation of new specificities in these receptors. When this paper was in review, we were very lucky that a real answer, so we did all of this without knowing the real answer, what the ligand binding sites actually are in any of these receptors. A real answer came out for one of the Arabidopsis NLRs called RPP1, and this is this loose enrich repeat module from it, and it's binding this effector called ATR1, and the residues in blue, all of the residues with side chains are those catching the effectors, And the residues in blue are those that we could recall with our entropy analysis, computational analysis, computationally. So overall, we can recall over 75% of the residues constituting a ligand binding site using this diversity metric. That's as good as antibody binding site prediction. So we're quite happy. And but using the structure, we could actually estimate precision and recall. What we also observed is that because for Arabidopsis, there's so much functional data for each of these receptors, what type of effectors they might be recognizing, the model of how they're recognizing it, that this indirect receptors that work through indirect recognition, monitoring a host protein targeted by effectors have low entropy. Makes sense, right? You're monitoring your own protein, not changing, you don't need to evolve away. Receptors with integrated domains, where this module is integrated, the beta gets, again, low entropy. But these receptors that are known to directly bind the ligand, so they act through direct recognition, they have these signatures of high entropy. And so we think now that using this comparative genomic and diversity metric analysis, and we've done it now, again, like for crops, right? We can pick out which of these receptors would be directly tracking the pathogens versus those that functionally work through other methods. Oops. And we can also predict ligand binding sites for those receptors. Now what? Well, last 5 minutes, I can tell you the first step that we're taking that are published on the using this information in engineering, in protein engineering of this immune receptors. And the proof of concept, and so the rationale there, if we could make synthetic receptors to recognize the structure, the pathogen structures, it will be much faster than the breeding that's going on right now. And the postdoc in the lab, she took a wheat NLR, a highly variable NLR. She also took another wheat NLR that also highly variable NLR. One of them is called SR50, and we know the effector at SR50. The other one, it's not an ortholog of SR50, it's called SR33. They're very different sequence-wise. We can compute entropy analysis for each individual orthogroups, and we can map it to the structure, right? Do what we did. And what she did then, she said, okay, if we take this predicted ligand binding site from SR50 and we create a synthetic construct where we just replace this 12 binding, it was initially 11 residues. I'll tell you about the one that became really important in this distant homolog. Can we change the specificity? Can we basically make this paralog recognize now the ligand? And the quick answer is yes. For those of you who like the data, we showed it by 2 different ways, right? Right now, using wheat protoplast and cell death assay to monitor activation on NLRs and wheat protoplasts, and Nicotiana benthamiana assay, where cell death here constitutes a signal active receptor, and the synthetic SR33 syn is able to recognize the presence of the effector. Now, I told you that this was 12 residues, and it's nice, but actually we failed in our first attempt. In the first attempt, we just took 11 residues that were highly variable, and we said, well, only the highly variable residues are sufficient. And the end result was autoactive receptor. So we broke something. We managed to break the system. And then Arginina and Kunyan, they were searching for the missing residue, and the missing residue turned out to be a lock between an LLR, one domain that binds the ligand, and the activator domain and BARC domain. And there's a co-evolution there's some changes occurring. It's not a highly variable residue. What we think, it's a residue that keeps the receptor in the off state. And so the binding site has to talk to the activator domain. The ligand comes in, knocks out, binds at the binding site, and is able to unlatch that lock. So it's a latch. So not only you need to transfer a binding site, you need to transfer a functional latch. And that was the 12 residue that gave us the full transfer of recognition specificity. Again, there is data showing that. And so what I told you for this quickly, the third story is that understanding natural diversity can be used to make synthetic designs. We are moving, like we're seeing how much we can push that boundary now with other projects, but at the very least, we showed that you can identify a disease-resistant gene in one species, so SR1533, or they come from rye, or from algae of Stalachy, so wild relatives. They were introgressed, right? But if you can identify a functional gene in the relatives, there is a potential to bioinformatically identify the important residues and make ideally, like a future vision, make a synthetic cassette, knock that into a paralogous copy, or like a blank copy basically in your crop. So again, wheat has 3,000 and a large 3,000 templates. If they don't work for your pathogen, if they're useless, they can become useful if you added them to gain the recognition of a pathogen. All right, I told you the acknowledgement already on the people who did the work, so I'm going to add with take-home messages. I think the natural diversity can reveal many different mechanisms of how new effectors and receptors arise. And for me, the future is to, one, understanding the biodiversity of how organisms, the outliers, how organisms can do things differently, and seeing if we can deploy it in crops. And second, the rational design of immune receptors. Thank you. And we do have 10 minutes left.
Really fantastic talk. I was wondering, in your system where you're able to modify the ligand binding pocket, but you have to change the latch as well, if you've been able to correlate amino acids in that latch region with amino acids in the ligand binding region, so you can predict what changes you might need to make to the latch, or not?
I'm not really yet. I think this is for, if we think about like completely novel binding specificities, that would be a lot of like build, test, fail, redesign, and we'll learn. Co-evolutionary, we couldn't observe like a strong enough signal for us to have a prediction. What Kunyang is doing right now, though, he is doing computational designs based on the based on the model and docking, and seeing what residues might be allowed and which residues might clash. So we can reduce that space. We can say what probably is not going to work, and making synthetics to test. Yeah, I think.
Yeah, great. Regarding the antimicrobial peptides, do you know what the type of antimicrobial peptides are they, and how many of them are they conserved across the plant?
Yes, so this is the MIAMP, the macadamia something. So they were discovered first in macadamia nut. And in macadamia nut, the MIAMP1, there's only one, and it is active against fungi. And it has basically also similarity to the yeast killer toxin structurally. In other plants, there are less of them. So Rhabdophis none, rice none, tomato none. There are, and in duckweeds, it's on the order of 30 to 50, and they're highly variable. So across different duckweed species, they're also differ very much in their sequence.
How long are they? Do they have more like cysteine?
Yes, they are cysteine. They have cysteines. They're about 10 to 15 kilodaltons. Aaron was able to purify a few of them already using a special E. coli strain that can help this fold. I would be thrilled to talk to you more about these assays, and we're happy to collaborate and like distribute them to test them against different, if you have a system to test them. And
so I have a question related to the first part of your talk. So for the effectors with similar structures, I'm wondering what's the idea? Are these effectors targeting similar proteins or diverse?
Diverse. I think it's the latter. I think what's happening is basically when a non-pathogenic fungus encounters the plants and tries to establish the infection, there's some sort of mechanism that leads to duplication of some secreted proteins that are useful for that. And then there is selection pressure for these to diversify we favor a hypothesis to then target anything they can in the plant, and then something of that would be useful and preserved. But the cost for fungi or eukaryotic pathogens to expand those is small. So unlike a virus where it needs to pack its genome and it's limited, here it's easy to have hundreds and thousands of secreted proteins. So I do think they target different proteins. They have different expression patterns.
Well, then I have two follow-up questions. One is, if they target different proteins, then, but you still, somehow evolve this same type of fold. Is that because there's something special about this fold that is easy to actually
yes,
kind of evolve from different, like origins?
I can tell you my favorite hypothesis. So, and I can tell you that reviewers asked us to do selection pressure analysis on different of these clusters. What we see is the residues that hold the fold are under negative selection, and the residues that are on the surface, they're either under neutral or positive selection. But there's basically within the protein, there's this combination of selection pressures of this. And your question was, what's special about the folds? Probably it's a good stable fold to, my hypothesis, to withstand diversifying the surface. It's one, that's what makes it useful. And then the second one maybe, but that I don't know. If you imagine a protein that naturally can target different things, like it's an enzyme, but it has lots of different substrates, right? So it's already a good platform for targeting things. So that's again, but that's now kind of, I have no thing. I'm just thinking like, well, you need a fold that's stable enough. That's okay, that's true. We even showing that. But maybe if you're starting with a protein that naturally is not a specialist protein, but a generalist protein, then it can be co-opted better to then target proteins from another organism.
Yeah, so then the question is, if you use the same approach, well, you must know that, in the host, right? If you look at their genomes, they must have proteins with similar fold, like like this are RNAs like domain. So what make those, what are those proteins? Effectors to some extent, or what makes them not functional as effectors?
If a host, so this actually, we did look at it, and actually this rarely happens. We see very few cases where we see a similarity between an effector fold and a host fold. But we did look at it. It's part of the table. I can tell you, Young looked at Arabidopsis, rice. I don't know if he methodically did for each pathogen, each host. I think he kind of gave up, not seeing much of a signal there with a few that were, but that was the data.
I was wondering if pathogens from multiple kingdoms secrete effectors that target hubs inside the plant cell, let's say the COP9 signalosome, would those effectors from these different pathogens potentially also converge at the structural level?
They don't. The current answer is maybe some of them would. So we did see one slide I took out at the time saying we did see examples of what we think is convergent evolution. Cases where it's really, we think evolutionary unrelated, but the effectors that they have the same fold, but I think you can target same protein very, very different ways. So for example, like bacterial type 3 secretion proteins, definitely they don't have similarity to fungal. And even across the fungi, we have very different folds. So again, I think like I cannot put the numbers on it. It would be really useful to look at it. Yeah.
We generally define the effectors like the protein molecules that have the secretory signalopeptides. So I want to know, like, do you know if there are incidents when the non-secretory factors, like the non-effectors protein, they gain the signalopeptides and become factors from non-effectors?
That's a great question. A reviewer asked us. We actually looked at it, and we were lucky because we were doing all of this analysis and structure predictions at the time when the kind of like a global structure, you go to, you know, Ensemble or EBI and you can get an unfold version for your protein that was not available. Now, at the time we were in review, that became available. So we took all of the proteomes for some of the species, and the reviewer asked, like, can you do it for one? And we were like, yeah, sure, we do it for like almost everything. And we looked for structural homologs of secreted proteins, especially, and we focus on highly expanded sets, these pathogen-specific highly expanded sets in the secretome. What we observed, because the reviewer asked, is that, do you actually have an expansion compared to non-secreted protein? Yes. Do you have a homolog in the non-secreted protein? A lot of times, actually, yes. And it's even a sequence homolog. So you can trace, like, but not for all of these highly expanded folds, but for a few. And it's like you can have 50 or 100 expanded members secreted, and there will be like 1 or 2 non-secreted. But they are homologs, they're related usually by sequence.
Okay, so
But to caveat though, Kunyang thinks it's caveat though, that the caveat can be gene model, that these, what we call non-secreted, they just, the signal peptide might not be predicted correctly, or gene model might be not correct. So we cannot exclude the possibility that they are actually secreted as well. We call them non-secreted based on current gene models. Big caveat. So
another like related question. So are there any chances of like gaining these signalopeptides from other organisms like parasites or like microbes, or they can only be gained from the same individual?
no. Yeah, I don't know enough about signal peptides. Yeah.
So I have a question about this. Yeah, my voice is pretty loud, so I think it'll be all right. I have a question about this sort of friend versus foe story, which is really cool. And this is going to show my ignorance of Pseudomonas, probably.
Go ahead, mine too, probably.
So first question I was wondering about is, how does the friend avoid immune response or recognition?
We think AMPs.
Okay.
I think that's where AMPs might be important. They're good enough to like, yeah. Okay, go ahead.
Okay, yeah, that was the first thing. And second, what do you think the benefit that the friend gets from?
Yeah, so that's a good question. So to answer the first question, we think it's a tripartite interaction, right? And so I think the resistance to AMPs are partial, right? Like AMPs can maybe keep some sort of level and have, that's again hand waving, right? Right. So well, talk to me 2 years from now, 5 years from now, but maybe they are involved. So the host, the friend is resistant to whatever mechanism the host is using. And what does the friend gain? Nutrients, probably. I don't know. I mean, like what can a niche in a pond that is non-competitive and has nutrients? Competitive, but meaning like you're not in the your soup, right? Like if you're in a pond, you're soup of organisms.
Yeah. It's just cool to think about like what's the benefit of being a pathogen versus a friend, and
exactly.
what are the relative
Yeah.
benefits?
Okay, any more questions? Okay, let's thank you. Thank you, everyone. Okay,
Yeah, I'll let you stop the Zoom then.
Okay.
Stop.
Thanks.
Yeah,
That was
thanks for
great.
such a.
Thank you.
Yeah.
And I don't know if I'm seeing later.